So please let me know when I shall start. Yeah. Thank you.
Yes, sir, we will inform you.
Yeah, no
Assalamualaikum,
problem.
everyone. Um, Miss Ikra, are you there?
Yes, ma'am, I'm here. Should I start?
Yes, please take start.
All right. Okay. Assalamu alaikum, everyone, and a very warm welcome to all of you joining us live today. We are truly excited to have such a wonderful audience with us. My name is Ikra Faisal, and I'll be your host for today's session. Thank you for being part of this important and forward looking discussion. Before we begin, I'd like to briefly introduce the platform behind today's event. This symposium is organized by VitaNova, a scientific and educational platform committed to promoting research, innovation, and meaningful discussions in the field of life sciences. VitaNova aims to connect researchers, students, and professionals from around the world to share knowledge and work together toward real world solutions. As you know, behind every successful event is a dedicated team. I'd like to acknowledge Mr. Ghulam Murtaza, CEO of Dr. Vitanova, whose vision and leadership continue to drive initiatives like this symposium. I would also like to extend our sincere appreciation to Dr. Shehzad Zafar, Senior Vice President of Vitanova. Additionally, I'd like to recognize Ms. Hafsa Sarfraz, Vice President of Vitanova, for her dedication and efforts in ensuring the smooth execution of today's session. A big thank you to the entire Vitanova team for making this event possible. Now, coming to today's session, we are delighted to welcome you all to the International Symposium on Transforming Crop Genetic Resources for Changing Climate. In today's rapidly changing climate, ensuring food security has become more critical than ever. This symposium brings together science, innovation, and policy to explore how crop genetics can help us meet these global challenges. We are truly honored to have two highly respected experts with us today. First, we have Professor Dr. Wilson Thaulin Yong from the Biotechnology Research Institute of Malaysia. Professor Yong will share cutting edge insights on gene editing technologies, particularly CRISPR-Cas and their role in sustainable agriculture and human health. Joining him, we have Dr. Wazir Ali from Shaheed Benazir Bhutto University, Pakistan. Dr. Wazir will highlight policies that support climate resilient crop development, emphasizing how strong policies frameworks can complement scientific advancements. We are grateful to both speakers for taking time to be with us today. We encourage all participants to actively engage during the session. Please feel free to drop your questions in the chat box as the talk continues. We will address them during the question and answer segment at the end. And as for your attendance, you can write your name in the live chat. So, without any further delay, let's dive into today's discussion. It is my great pleasure to invite our first speaker, Professor Dr. Wilson Thaulin Yong, to begin his presentation. Over to you, sir, and welcome once again to everyone joining us today.
Actually, sir, you are not audible.
Hello.
Sir,
Can
you
you
can
hear me?
start now.
Can you hear me now?
Yes, sir, you are audible.
Okay. All right. So thank you, MC. Yeah. So, dear all distinguished guests and also respected participants. So thank you for joining this session. My name is Wilson Yong, and I'm with the Biotechnology Research Institute, University Malaysia Sabah, where my work focuses on agricultural biotechnology and plant molecular biology. So it is a great honor to speak at this international symposium on transforming crop genetic resources for a changing climate. So I would like to sincerely thank the organizers, so the Vita Nova International Alliance for Sciences and Shahid Benazir Bhutto University for the kind invitation. So today I will present my work under the theme Transforming Bioresources for Climate Resilient Agriculture. So this presentation will highlight how we integrate crop genetic resources and the aquatic biomass and biotechnology to address the pressing challenges posed by climate change. So the work I will share is grounded in laboratory-based research, system development, and the field experience that I conduct in University Malaysia Sabah, often in collaboration with my industry partners and also the local communities. Next slide. So, agriculture today is facing unprecedented pressure from multiple directions. So, food security is increasingly threatened by population growth combined with climate instability. Climate change brings unpredictable stressors such as drought, flooding, rising temperature, and increasing pest and disease outbreaks. At the same time, sustainability demands that we reduce environmental impact while maintaining productivity. So agricultural biotechnology offers integrated solution to these challenges by enabling adaptation, resilience, and also efficient resource use. So our work in University Malaysia Sabah aligns strongly with the Sustainable Development Goals, particularly zero hunger, good health and well-being, responsible consumption and production, and climate action, and life on land and below water. So this global context is why our research focuses on bioresources as the adaptive assets rather than single crop or technologies. We look into the overall combination of the bioresources. Next slide. So with this background, today's presentation follows an integrated pathway. So I will first discuss plant tissue culture, which serves as the foundation for safeguarding crop genetic resources under climate stress. Then I will move on to seaweed biomass, a highly climate resilient aquatic bioresource supporting food security and aquaculture. Next, I will also introduce a microalgae, highlighting their role in nutrition, energy, and environmental management. Following this, I will also present enzymatic biotransformation, which adds value to the natural bioresources. And finally, I will also outline collaboration opportunities to scale this innovation beyond the laboratory. Next slide. So the previous one, okay. All right, plant tissue culture for climate resilience. So climate change magnifies long-standing weaknesses in agriculture. Traditional farming often suffer from inconsistent quality, disease vulnerability, and also genetic erosion. And plant tissue culture allow us to address this issue at the genetic resource level. So in our laboratory, tissue culture is used not only for rapid propagation, but also for genetic stabilization and conservation. So by producing uniform and disease-free planting materials, we improve the resilience and also reduce climate-related risks in agriculture. So this transforms tissue culture into a core climate adaptation strategy rather than only a propagation tool. Next slide, please. So bamboo is a strong example of how genetic resources can support a climate solution. So it grows rapidly, stores carbon efficiently, and also contributes to land rehabilitation. However, large-scale use is limited by inconsistent planting material quality. So through tissue culture work in our lab, So we develop genetically uniform bamboo plantlets, and these plantlets are suitable for reforestation, ecological restoration, and also climate mitigation projects. So here, biotechnology directly supports carbon sequestration, biotechnology recovery, and also sustainable resource supply. Next slide. So ginger production also is increasingly affected by a climate-related disease pressure. Warmer temperatures and also increased soil moisture promote the spread of soil-borne pathogens, resulting in severe yield losses. Conventional farming methods struggle to manage these challenges effectively. So in our laboratory, we collaborate with the local farmer. We use tissue culture techniques to produce pathogen-free ginger planting materials. And these plants show higher germination rates, improved vigor, and also more stable field performance. So this would also demonstrate that how biotechnology can reduce climate-induced disease risk and also support productivity and farmer resilience. Next slide, please. So coffee is among the crops that are most sensitive to climate variability. Temperature fluctuation affect flowering, fruit development, and also bean quality. And this will directly impact the market value. Maintaining consistency under this condition is a major challenge. And our tissue culture work focuses on producing uniform coffee planting materials with stable quality traits. And the lab-grown seedlings demonstrated consistent growth and adaptability under the variable field conditions. And this approach supports the long-term climate adaptation strategies for high-value perennial crops. Yep, next slide. So corn, corn is another plant that plays a central role in global food security. Climate change intensifies the pest pressure, disease incident, and drought stress, threatening the stable production of the corn. And yield instability can also have serious consequences for food systems. So through tissue culture, we support the propagation of more resilient planting materials. And this approach emphasizes yield stability and stress tolerance rather than to maximize the yield alone. So these strategies are essential for protecting the stable crops under the climate uncertainty. Next slide, please. The medicinal herbs. Okay, medicinal herbs are particularly vulnerable to climate change and in the local community also facing over-exploitation, habitat loss, over-harvesting, and environmental stress threaten many of valuable medicinal herb species. So conservation strategies must balance protection with sustainable use. So using tissue culture, we can conserve the endangered plants while ensuring consistent bioactive compound production or the materials production. And this supports research, commercialization of the products, and also biodiversity conservation simultaneously. So this is how the biotechnology can link the genetic conservation with bioeconomic value creation. Next slide, please. So now we move from a land-based crop system to aquatic bioresources, which are increasingly important under the climate change. So seaweed stand out as one of the most climate resilient forms of biomass. So it requires no fresh water, no arable land, and no chemical fertilizer, as like the land-based crops. And also, seaweeds are actively supporting a coastal ecosystem. However, climate stress, pollution, and disease outbreaks have significantly reduced the availability of healthy seedlings in many farming areas throughout the world. So this instability directly threatens the aquaculture productivity and also the coastal livelihoods. So our research focuses on stabilizing seedling supply as the first step toward the sustainable seaweed-based food system. So in this context, seaweed is not just a crop, but also a nature-based solution for climate resilience. So building on this, seaweed availability becomes the critical bottleneck. Seaweed farming increasingly faces climate-driven disease outbreaks and environmental instability. So when seedlings are unreliable, industry growth is slow, and the farmer income also becomes unpredictable. So to address this, we applied tissue culture techniques to improve genetic stability and survival rates. And through this approach, we achieve more consistent and resilient seedling performance. And therefore, this seedling production formed the foundation of aquaculture systems, linking the laboratory innovation to field sustainability. Next slide, please. So once the propagation is achieved, the next challenge is to optimize the system. So in our work, okay, we systematically optimize the parameters such as light intensity, temperature, and nutrient composition under control conditions to produce good quality seaweed seedlings for the farmer. So these parameters significantly influence the growth rate, survival, and also the seedling quality. We also develop a water recirculation system to improve the resource efficiency and to reduce waste, to minimize environmental impact while maintaining high productivity. So this system level innovation are essential for scaling seedling production in a climate smart and sustainable manner, okay, by producing high quality seedlings for the farmers. All right. So this slide, okay, show the quality of the micropropagated or tissue cultured seaweed. Okay. As the production system become more intensive, so quality assurance become increasingly critical, especially under the climate variability. So in our system-based trials, the system produced seedlings show faster growth and also higher nutritional content as compared to the conventional farm-propagated seaweeds. So importantly, the cultivation cycle of the seaweed farming was shortened from 45 days to approximately 30 days by using our system-produced seedlings. And the standardized cultivation protocols and regular quality control ensure the consistency in the products and to be applied in food, feed, or pharmaceutical applications. And this consistency builds the market confidence and also support wider industrial adoption. Next slide, please. So beyond the production efficiency, the real impact of seaweed lies in its application across food and the feed systems. Seaweed contributes directly to a climate resilient food system through its rich and diverse nutritional profile. It provides essential minerals, high quality proteins, and also functional compounds that support human nutrition and health. So when incorporated into animal or aquaculture feed, seaweed helps reduce the environmental footprint of production systems while also improving the feed efficiency. So in our work, okay, we observe that improved growth performance and overall health in aquatic species such as fish and shellfish by using seaweed-based feed. And this positions seaweed as both a nutritional solution and also an environmental intervention in climate resilient agriculture and aquaculture systems. Next slide. So now we move on to the microalgae. So microalgae can operate at even a more intensive biological scale because microalgae offer integrated solutions to multiple climate-related challenges simultaneously. They grow rapidly, efficiently capture carbon dioxide, and generate high-value biomass with diverse applications. So as urbanization and industrialization continue to increase pollution level and energy demand, microalgae provides sustainable responses. So our research explores the use of microalgae in food, in animal feed, in bioenergy and also environmental wastewater remediation. So in this context, microalgae function as a secure bioresource platform, linking the climate change mitigation with resource recovery and sustainable production systems. Next slide. So a key advantage of microalgae lies in their extraordinary biodiversity, much of which remains largely untapped. Bioprospecting allows us to identify strains that are naturally adapted to environmental stress and variable growth conditions. So these stress-tolerant strains often produce variable nutrients or bioactive compounds when grown under the challenging environments. So in our lab, we focus on systematic screening and selection of microalgae with a clear industrial relevance to ensure that scientific discovery is closely aligned with the downstream applications. So this targeted bioprospecting supports sustainable and climate resilient innovation, particularly in the development of bio-based products for food, animal feed, and also health-related industries. So energy transition is another critical dimension of climate resilience. So continued reliance on fossil fuels accelerates the greenhouse gas emissions and also climate change. Microalgae offer renewable and sustainable alternative due to their rapid lipid accumulation, high productivity, and also ability to grow on non-arable land. Beyond biofuel production, microalgae can also be used to generate bioplastics, biohydrogen, and also a range of specialty chemicals such as pigments, carotenoids, antioxidants, and also biosurfactants. So these bioproducts contribute to reducing carbon emissions and dependence on fossil-based resources. So in this context, bioresources play a role not only in strengthening food system, but also in advancing energy security and material sustainability within a low carbon and circular bioeconomy. Next slide. So another major challenge intensified by climate change is the protein security. So conventional protein sources are increasingly constrained by limitation in the land availability for farming, water resources, and also feed inputs. So high protein microalgae offer a sustainable and reliable alternative to traditional protein source. By requiring only minimal land and water, microalgae produce sustainable products, reduce pressure on agricultural system, while providing consistent and high quality nutritional profiles. So their inclusion in food and feed applications support a more resilient and diversify nutrient strategies for both humans and animals. So in this way, microalgae contribute to food security at both the quantity and quality level, strengthening the nutrition system under the climate uncertainty. Next slide. So beyond biomass production, microalgae also enable innovative solutions for environmental management. Industrial wastewater poses serious environmental risks if not properly treated before discharge. So in our study, microalgae have been applied to treat palm oil meal effluent, effectively removing excess nutrients and reducing pollution lots prior to release into the environment. So at the same time, this process generates variable algae biomass that can be further utilized for applications such as biofertilizer, feed ingredients, or as a source of bioactive compounds. So this integrated approach closes the loop between the waste management and resource generation, providing a practical demonstration of circular bioeconomy principles in action and support more sustainable industrial practice. Next. All right. So we move on to enzymatic biotransformation, because up to this point, the focus has been on the production and optimization of bioresources. So the next step is the value addition, using enzymatic biotransformation. So enzymatic biotransformation allows us to modify natural compounds into more functional forms. So by using microbial enzymes, so we enhance the compounds derived from plants, from seaweed, and from microalgae in a targeted and environmentally friendly manner. So this approach increases bioactivity and functionality without relying on harsh chemical processes, thereby maintaining the environmental sustainability. So importantly, enzymatic biotransformation complements the upstream bioresource production and maximize the overall value derived from bioresources. Next slide. So many naturally occurring compounds exhibit promising biological properties, but often show limited effectiveness or stability in their native form. So through enzymatic transformation, we generate novel bioactive compounds with enhanced functionality, improved bioavailability, or greater stability. So these transformed compounds have strong potential applications in pharmaceuticals, nutraceuticals, and functional foods, including areas such as antioxidant enhancement, metabolic health support, and also cosmetic or skin-related applications. So this step effectively bridges the fundamental biological research with industry-driven innovation, accelerating the translation of laboratory discovery into practical and market-relevant products. Next slide. So looking ahead, so our research focuses on combining enzyme to achieve enzyme synergy and process optimization to further enhance the functionality of natural compounds derived from plants, seaweeds, or microalgae. So we work with enzymes such as amylosucrase or tyrosinase to selectively transform plant and algae-derived compounds. For example, amylosucrase can introduce glycosyl groups, improving solubility and stability, while tyrosinase can facilitate oxidative modification, leading to the formation of functional phenolic derivative with enhanced bioactivity. So by combining these enzymes, we create opportunity to improve reaction efficiency and tailor functional properties. So future evaluation of the novel compounds focus on antioxidant, anti-cancer, and also cosmetic-related bioactivities, moving this work closer to real-world applications and commercialization. Next slide. So to conclude the scientific narrative, so our work demonstrates an integrated bioresource strategy spanning plant tissue culture, seaweed, microalgae, and also enzymatic biotransformation. So together, these approaches safeguard and transform biological resources from genetic conservation to value-added product development. So this integrated framework shows a significant potential for climate resilient applications, enabling a stable production system under environmental stress while enhancing functionality and efficiency. Plant tissue culture strengthens crop genetic resources, while seaweed and microalgae provide scalable, low carbon biomass for food, feed, energy, and also environmental remediation. So by combining these bioresources with enzymatic biotransformation, so we support sustainable agriculture and bio-based industries grounded in practical system and validated laboratory outcomes. So moving forward, yeah, collaboration is essential to scale impact, okay, and translate research into real-world solutions. So we in University Malaysia Sabah, we invite partnerships to produce high-value bioresources using tissue culture, ensuring a stable, reliant, and a scalable plant supply. Develop novel compounds through biotransformation, creating adding value from plant, algae, and microbial resources. Establish strong quality and safety frameworks, supporting regulatory compliance and building market confidence. And lastly, accelerate innovation across agriculture and biotechnology from laboratory discovery to industrial application. So through a strategic collaboration, we can transform scientific advances into practical solutions that address climate resilience, sustainability, and industry needs. So I would like to acknowledge University Malaysia Sabah, our funding agencies, industry partners, community collaborators, and my research team. And these achievements are the results of the collective effort, shared vision, and also long-term commitment. So thank you very much for your attention, and I look forward for your comments and if possible, future collaboration as we continue transforming bioresources for climate resilient agriculture. Thank you.
Thank you so much, Professor Dr. Wilson, Thau, and Yong, for your insightful and impactful presentation on gene editing technologies and their potential role in developing climate resilient crops. Before we move on to our next speaker, we invite the audience to share any questions related to Dr. Wilson's talk in the chat box. We'll take a few of them during question and answer segments.
Yeah, thank you so much, respected Professor Dr. Wilson Thailand Jung. I really appreciate your time, and we honor to have you here uh, for beautiful presentation.
Thank you for the invitation. Yeah, thank you for inviting me to share my research in the university.
Uh, Dear participant, if you have any question, you can write in chat box. Our host will read out your question, and dear Professor Dr. Wilson will answer your question if you have.
So by the way, the pictures on the slide is the pictures of the University Malaysia, the campus of University Malaysia Sabah. So we welcome you to visit us, yeah, and to have a long-term collaboration with us. Yeah, thank you.
Yeah, thank you so much. So we will also discuss about collaboration, future, how we can collaborate. Uh, we are initiating a project about plant sciences, especially genetic engineering.
Thank you.
So
Thank you. Good.
we want to also collaborate. And here with us, Dr. Wazir Ali from Shahid Benazir Bhutto University in Pakistan. So our next speaker. So I want to respected Professor Dr. Wilson. I want to meet you with Dr. Wazir Ali. Is a distinguished international scientist in plant protection. Yeah,
Yeah, sure.
Mr. Mansoor, please join the Mr. Dr. Wazir. Mr. Mansoor, kindly join the Dr. Wazir Ali. Hello, Dr. Wazir. Dr. Wazir Ali, Metlo, can you hear me? Just 2 minutes. Soon he is joining. Yeah. Mr. Mansoor, kindly join the Dr. Wazir Ali Metlo. If anybody have any question, you can write till 4 5. Miss Hafsa, kindly upload the next presentation. Thank you so much. Uh, Respected Professor Dr. Wilson Thau Yong, uh, just within 2 minutes, I just want to meet you with Dr. Wazir Ali, and then we will conclude your session. Thank you so much. Mr. Mansoor, is Dr. Wazir here?
No, sir. He's not available right now.
Ms. Safsa, kindly upload the next presentation.
Okay, sir, sure.
Hello.
Hello, sir.
Yes. Yes. Assalamu alaikum.
Walaikum salam, sir.
Yes, Dr. Wazir Ali speaking. Did you listen my voice clear?
Yes, sir, your voice is clear. We can listen you.
Yes. Can I start my presentation?
Yes, sir, just give me 1 minute. Miss Ikra,
Uh,
are you there?
Yes, I'm here.
Okay, please
yes.
welcome doctor and then take start.
All right, now moving forward with today's session, it's a pleasure to introduce our next speaker. We are honored to welcome Dr. Wazir Ali from Shahid Benazir Bhutto University, Pakistan, who will be sharing valuable insights on policies supporting climate resilient in data palm development. Dr. Wazir, you can now proceed with your presentation. Thank
Uh,
you.
just,
Okay.
just, just, just wait a minute, please. I
Okay.
just want to, respected sir, I want to meet you with Dr. Wilson Yong, is a professor in University Malaysia Sabah. Uh, yeah, he is here.
Hi, Dr. Wazir. Nice to meet you
Yes.
here.
Uh, yes. Thank you. Thank you. Thank you very much, sir.
Yeah, yeah.
Thank you very much, sir.
Hopefully we can have some
Nice,
collaboration.
nice
Yeah,
presentation, Dr.
in the
Saab.
future. Yeah, good to see you. Yeah, looking forward to your presentation.
Thank you.
Thank you so much, respected professors, Dr. Wazir Ali and Dr. Wilson Thau. I really appreciate. And Dr. Wazir Ali, please, you can continue.
Yes, sir. Thank you very much for giving me an opportunity to deliver on the lecture on the policy supporting climate resilience and date palm development. Next, please share a slide. Next, please.
Yes, sir, slides are shared.
Yes,
Please move the slide.
but cannot show here.
Actually, at our side, slides are shared.
Yes. Okay, okay, okay. If we introduction, if we know about the date palm Phonetectalifera belongs to the family Palmaceae, which is a large family and comprising about the 200 genera and 1,500 species. It is a major fruit crop of arid, desert, and tropical and subtropical regions of the world, including the Middle East, North Africa, and South Asia. The main date producing countries are Egypt, Iran, Saudi Arabia, Pakistan, United Arab, United Arab Emirates, Oman, and Libya. In these countries, the dates play a major role, significant role in the socio-economic stability of the countries and contribute to the national income and the date producing areas in the world. Next, please. Here is the palm, the ancient evidence of the date palm. If we look into the Mohenjo-daro, the date palm was cultivated the 5,000 years ago. These are the archaeological evidence of date palm in Pakistan. Next, please. If we look into the problem of the date palm, the climate change and date palm cultivation. So what are the climate change? The Pakistan, if you look into the Pakistan, among the world leading producer of date palm, with major orchards concerned in Sindh, Khyber Pakhtunkhwa, southern Punjab, and parts of the Khyber Pakhtunkhwa. If we look into the Sindh, the Khairpur and Sakhars is the biodiversity center of the date palm, having more than 300 varieties are grown. The intensified climate risk heat waves, heat wave kinetic and extreme monsoon rainfalls, drought, salinity, flood, disease, and pest are reshaping the production risk along the entire value chain from the flowering to the pollination of the dying. These are the major problems of the date palm. One is the climate change, and other is the sudden decline disease and pest, laser death moth. These are the major issues of the date palm. Next, please. These are the impact of the climate change on date palm. The people don't know about the symptomatology of the climate change. Symptomatology is the insect and pest, and symptomatology of the fungal disease, nematode disease, and viral disease. So these are the symptoms of the climate change. The date palm trees are drying. Next, please. These are the impact of climate change. The climate change on date palm cultivation. The climate change affecting the date palm in Pakistan. The multiples in the way in terms of production, yield reduce. The some major impact are disease under the rising of the temperature, which put the negative impact of the palms and heat waves. Erupted monsoon rain. When the date palm ripening, the monsoon rains harvest all fruits of the date palm and it puts a negative impact of the farming community in Pakistan. Drought and water scarcity. Drought and water scarcity is the major problems of the date palm. The palms are drying it. The soil and water salinity and next flood and waterlogging increase the pest and disease incidence in the date palm. These are the major issues which reduction of the date production in Pakistan as well as in the other countries of date producing areas in the world. Next, please. These are the major date producing areas in the world: Saudi Arabia, Egypt, Iran, Pakistan, Tunisia. These are the date producing world. These are the map and the produce of the areas in the world. Next, please. Cultivation of date palm in Pakistan. The date palm cultivation in Pakistan is concentrated in the four provinces and forms an importance of the where the most important date palm genotypes are cultivated in 2004. The industry supported by gross domestic products and many people say numerous farming and traders can millions of the rupees earn millions of the rupees from date production and related to the business. They also supported the processors and exporters. The dates farming support the farming and others. These are the main producing areas of Pakistan, Sindh, Sakhar and Khairpur, Punjab, Ramyar Khan, Bahawalpur, and southern parts of the Punjab, Khyber Pakhtunkhwa, and Balochistan. Mashkal sites are the Balochistan. These are the areas producing of the date palm. If we know about the Sindhu, the date palm in Sindhu, date palm in Sindhu is mainly concentrated in the district Kharpur, and it is the biodiversity centers having the more than 300 varieties are grown. The date palm is the major cash crop of the Kharpur. The people earn millions of rupees from this crop, and the district is the one of the leading date producing regions of the Pakistan. Next, please. Field survey of date palm disease. If we look into the problems of the date palms, we can also discuss the problem of date palm, the climate change, and others the red palm bevel. And in Sindh, particularly in the Khairpur, the sudden decline disease is a real threat of date palm decline. In this date palm research institute, Shah Abdul University, Khairpur, conducted field surveys in the Khairpur, indicated the most of the date palm orchards affected by a sudden decline disease caused by the fusarium, caused by soil-borne fungi attacks, which poses a serious threat to the date palm industry. In this region, the farming community is facing a real threat, and the symptoms are similar to those of the bio disease caused by the fusarium oxide. If we know about the biot is the one of the most destructive soil-borne disease of Algeria, where the 3 million date palm trees in Algeria have been destroyed, and 12 million in Morocco. If we know the condition of the Morocco, Morocco was the first the importers of the exporter of the dates, and now the importer. The still the African countries of the Morocco, Algeria, and they suspected in the bio disease, same like sudden decline disease occur in the Pakistan, is the major disease. The symptoms similar to well-known bio disease of date palm caused by Fusarium oxysporum. Next, please. The impact of sudden decline disease at the Khairpur, the sudden decline disease has destroyed the hundreds of the orchards and dispersed the trees at the areas. The number of infected trees increasing day by day. Some areas are considered infection spots like Diplodia disease, leaf spot disease of the date palm. Now, sudden decline disease has become a serious threat to the date palm in Sindh, particularly in the Khairpur district. Next, please. Similar symptoms in different countries. The decline disease of date palm has also been reported from different date palm growing areas of the world. The Egypt, Iraq, Iran, Saudi Arabia, Libya, Tunisia, Oman, and Pakistan. Where the Saran is also reported from the Iraq 2001, Rashid and Abdullah Hafiz reported from the Egypt, where the Abdallah is a reported decline of date palm in 2000 in Saudi Arabia. Mansouri is also reported for 2003 in Iran, where the Metlo is also reported 2009 in Pakistan's Khairpur, Sindh, Pakistan. Next, please. The field survey of date palm sudden decline disease. The major disease, the maximum disease incidence was recorded in Nurpur, Babarlo, and where the minimum disease incidence was recorded in Shadi Shahid and Nunari. The northern part of the Khairpur is more susceptible than the southern part of the Khairpur. Next, please. These are the symptoms of the sudden decline disease. The primary symptoms appears on the ash gray color. Some spine situated on the side of the leaf become white, Then the disease progresses from the base to the apex. After one side has been affected, the after the one side has been affected, the whitening begins on the other side. These are the scientists late Professor Dr. Adil Ahmed come from the Egypt and visited the infected orchards of Khairpur. Next, please. Material methods means how the research conducted in the date palm research in Shah Abdul University Khairpur, which is a collaboration of Institute of Horticultural Science, Cairo University, Egypt. The sample belonging to the research laboratory for the isolation and identification of the pathogen, real pathogens responsible for decline of date palm in Pakistan. Next, please. The collection of the samples, the 70 different locations of Khairpur collected the sample and disease incidence and disease severity was recorded and collected from the different samples and bring to the research laboratory at Shah Abdul University, Khairpur. Next, please. These are the sample collection identification with the team of the Date Palm Research Institute, Shah Abdul University, Khairpur, Department of the Microbiology, and the team of the National Agriculture Research Center, Islamabad, came to visit to the infected orchards and collected a sample for the identification of the pathogens for decline of date palm in Pakistan. Next, please. Results. Result the six different fungi, namely Fusarum, Solani, Forma Euclidum, Helminthosporium, Sativum, Alternaria, Alternata, Aspergillus niger, and Penicillium chrysogenum were isolated from different parts of the date palm. These are the frequency uh, of soil inoculation. The data frequency of isolated fungi from the roots, leaves, and difference were recorded on the following formula. The colony, the number of the colony divided total number of pieces studied. The which species the more frequently for the decline of date palm. Next, please. The pathogeny test of different fungi, the pathogeny test was conducted on the three most commercial important varieties of the date palm, Asir, Fasuli, and Karbalan, and three different species were inoculated in the healthy seedlings of the date palm to showing the same symptoms and showing the taste of the pathogenicity test and for reconfirmation either is the palm is infected of the soil-borne pathogens or in other pathogens. Next, please. These are the soil inoculation methods. In soil inoculation methods, require the spore suspension of test pathogens were incorporated in the pot soil. After one or seedling transfer, the seedling grown in soil emanated with only distilled sterile water without pathogen survey as a control. The three methods were applied: soil inoculation method, spraying inoculation method, and stem inoculation method. Which methods is the more frequented in the pathogenicity test? Next, please. Stem inoculation methods. The quantity of spore suspension were injected in the stem of the date palm seedling with the help of the syringe in control plants. 5 ml distilled sterilized water was injected in the stem of the date palm seedling. Next, please. Spraying inoculation method, spore suspension of the each pathogen was inoculated by spraying approximately 5 ml plant seedling. Spraying with distilled sterile water were served as a control. Next, please. After 2 weeks of inoculation, the plants were monitored for the development of the disease symptoms. After 45 days, the inoculation, the seedling were uprooted, and data was recorded on the seedling mortality, disease severity, and recovery of the pathogen. The experiment was arranged, randomized, complete, block design with the three replications. Next, re-isolations in plant pathology. And in the pathogen test, the Koch-Pasteur rules were applied, and the re-isolation was also done from the inoculated and uninoculated plants, described above to confirm the pathogen nature of the test fungi. The necessary for inoculated from fungus was recorded, and the maximum person was calculated with the help of the following formula. Next, the fungi isolated from different varieties and their frequency. The maximum fungi, the Fusarum solani was isolated from maximum frequency on the 57 on the acyl and Fasuri on the 45% and the Carboline and Carboline on the 15%. It means the acyl is the most susceptible of the fungus pathogen followed by Fasuli and Carboline. Next, the recovery of the different soil inoculated fungi, the soil inoculation methods is the more frequented. The 100% plant mortality in fasuli, in acyl varieties, fasuli, carboline and fasuli. Next, the disease severity of different fungi, soil inoculation, the disease severity on the 100% of the soil inoculation methods, followed by soil inoculation methods in acyl, fasuli, and carboline. Next, please. Conclusion. Conclusion, it means the Fusarum solani fungus was the predominant fungus isolated by very high frequency, followed by Phoma eucalyptum and Helminthosporum sativum. The pathogen test of date palm seedling were carried down the most commonly growing date palm variety of seal fasculi and carboland by three methods of inoculation. Among three methods of inoculation, soil inoculation method was also very high frequency, effective in terms of recovery of inoculated pathogen. Fusarum solani was recorded in a very high frequencies from 98 to 100% from inoculated plants. Next, conclusion from the climate resilient germplasm and a varietal improvement. It means the policy support for the drought, heat, and salinity tolerant date palm varieties through the Public Research Institute. The establish the if the palm is infected from the climate, the farming community directly related concern with the research institute. They established the National Date Palm Gene Bank to conserve the local and climate resilient cultivars. Encourage the tissue culture and certify the nursery where the Dr. Wilson is also give the detailed presentation and the tissue cultures and crop resilient. Okay, thank you very much. Thank you very much for the team of the Vitanova International Science for conducting the wonderful session. Uh, Wonderful session. And thanks for all team.
Thank you so
Any
much, sir,
question?
for such an insightful discussion. Yes, I had seen some questions. Should I read for you, or you can take from the chat? Actually, I had seen question for Dr. Wilson too. Guys, I request you all to type your questions again because due to comments, it's go behind, so it's difficult to find each question. So I request you all to type your questions again. Miss Ikra, are you there?
Yes.
Here is a question. How re-isolation the pathogen method in transforming crop technology into the advanced modern advancement technology?
Thank you very much. Very good questions, madam. First of all, we have collected the sample from the field and bring to the research laboratories and prepare the PDA medium, potato dextrose agar medium से and make the 1000 ml से and transfer the disease specimen से. After 5 days, the fungal colonies were appeared and and we can transfer to the slide for microscope under slide under the microscope, okay. And we can identify on the morphology colony basis and different colony colors conidia basis and these are the isolation and identifications of the fungi. Next question.
Okay, sir. Thank you so much for answering. Here is the next question. As date palm weevil is a major pest of date palm, how to eradicate this pest effectively?
Yes, madam. Repeat again.
Okay, here is
Hello.
someone asking, sir, is I audible?
Yes. Next question,
Someone
please
is
repeat.
asking, okay, as date palm weevil is a major pest of date palm, how to eradicate this pest effectively?
Uh, as far as we have conducted the different experimental and chemicals in vitro applications for the control of the pathogens and as well as in vivo applications the applied of the different types of the chemicals of the different company like Beviston, Topson, uh, copper chloride, mancozeb, topson is the more frequent for reduction of the disease severities. The next palm trees give the fruit. If the palm is infected in the early stage of infection, the Beviston chemicals prove to be the highly effective against the pathogens.
Okay, sir,
Next,
thank you. Here is the next question. Someone is
yes.
asking that I did not understand when you were telling about how we inject it to stem while using syringe. Is this will be take properly and inside the plant? How we confirmed?
Uh, we have conducted the pathogeny test in different side. One is the soil inoculation methods, soil inoculation methods, and the seedling obtained from the date palm tissue culture laboratory for identified of the varieties, and in sterilized soil we applied and gave the five spore suspension in the soil and other injected plant through the injected method from the spore suspensions they give injected to the seedling of the palms and next to the spraying methods they give spraying on the upper side is spraying say of spore suspension of the pathogen to show the pathogen test and see the recovery of the pathogens which method is more frequent and the data indicated and result indicated that the soil inoculation method is the more frequented for reduction decline of the date palm. and the same pathogen, say Fusarum solani, was isolated. And this research was conducted in the Date Palm Research Institute, Shah Abdul University, Khairpur, and with collaboration of Institute of Horticulture, Cairo University, Egypt. These results is under supervision of foreign professor, late Dr. Adil, and sitting and the other team of the microbiology, Shah Abdul University, Khairpur.
Thank you so much, sir. Here is next question. How we trans how we transform the remote areas of date palm research to advanced technology reforms like in Sindh and Balochistan.
I think the uh, Date Palm Research Institute, Shah Abdul University, Khairpur, uh, conduct a very good research, and it is applied research. It has a very good tissue culture lab, and he has produced thousands of the hundreds of the date palms germplasm against the climate change and the and other components. It means very good research conducted in the Sindhu in the Khairpur, other side in the Balochistan. I can also visited the Balochistan, and next I also visited the city Karachi. So I think the work of the debris is more appreciable. Next,
Thank you
madam.
so much, sir, for answering. Here is the next question. To which part of the plant is injected mean to tissue or something else?
Stem injection methods, injection methods, injected stems.
Okay, sir. Thank you so much. I think all questions are considered. Guys, if you have any question, please write in the chat. And Dr. Wilson is also here. If you have questions from Dr. Wilson, you can also ask. Otherwise, he had shared
Okay,
his
okay,
email
okay,
in
okay,
chat.
okay, okay. I am ready every time. And the WhatsApp and the LinkedIn in groups, and inshallah I will replies with scientifically proved as far as my knowledge is concerned.
Okay, sir. Thank you so much for your concern.
Thank you, madam. Thank you for your patience and attention.
Okay. Guys, I think
Thank
questions are ended because further I cannot see any question. Here is someone is asking, what is epidemiology of fusarium in date palm?
epidemiology. The
Yes, sir.
Fusarum solani fungi is a soil-borne pathogen, and they remain in the debris of the plants. And the microcondia and the microcondia remain in the parenchyma and the collenchyma cells, and the conidia remains of the 8 years. If we uh, show in other crops, the conidia germinated and penetrate the root of the host plants, and stop the nutrients of the plants. The mycelium developed in the parenchymal vessels. After the develop the complete hyphae and the vessels, the plant don't take the nutrients and uptake the toxins of the pathogens. Then you can show the symptoms of the palms like yellow light. Did you get my point?
Yes, sir. Yes, sir. Thank you so much for answering. Uh, Guys, if you have still any questions, you can write in the chat. I am waiting for your side.
Okay, okay.
Yes, Kufaitullah, your question is answered. Sir has discussed it earlier. Yes, Aisha, please repeat your question because I did not get what did you ask. Okay, sir, someone is asking about re-isolation. Please explain it.
अच्छा, re-isolation. Hello,
Yes, sir.
hello.
Yes, sir.
Yeah, one can send me the chat on the re-isolation methods. Hello,
Yes, sir. You
hello.
are audible.
The re-isolation method, it means when you applied in the culture of the pathogens in the date palm seedling or in other plants, and you can show the and monitoring the disease development in the palms. The 45 days of the date palm seedlings were uprooted from the pot experiment and uprooted and show the same like procedures, prepare the medium, and then can you show the cut the infected part of the seedling and transfer the PDA medium. This is the re-isolation process. And same like check in the under the microscope and show the same like species of the Fusarum solani, Forma eucalyptus, Helminthosporosetium, Aspergillus. And here the only the Fusarum solani were inoculated on the three different methods of inoculations. So re-isolation is also same like the procedures, and it should necessary in the pathogeny test for the confirmation of the pathogen, either same pathogen or another pathogen. Did you get my point?
Yes, sir. Yes, sir. Thank you so much for answering. Here is next question. What is the definition of green world via climate policy and development?
What are the green word and climate? It means a climate resilience policy uh, show the hello. Climate resilience germ plasm are varietal improvement. It means you can develop in other resilient germ plasm against the climate change, and policy support for the drought, heat, salinity tolerant date palm variety through the public research institute, because the public research institute, the Ministry of Climate Change, give their policies and the climate resilient crop. Yeah, or you can establish the national date palm gene bank, which genotype you have collected from the gene bank and cultivated against the resilience of the crops, coast local and climate resilient cultivars. And these are encouraged, yeah, or you can also get from the tissue culture. And the Dr. Wilson is also give the detailed presentation on the tissue culture. Yeah, you can get certified varieties from the tissue cultures against the climate resilience. Hello.
Thank you so much, sir, for answering.
Okay, okay,
Uh,
okay.
Here is next question. Someone is asking, your institute in Sindh and it is an old university. People today not know about your institute and work. Why?
And it's not a old university. Shabdat University, Khairpur. Respected sir, Institute Shabdat University, Khairpur means it is a old university. People today not know about your institute and what. Wow. Yes, sir, Date Palm Research Institute, Shah Abdul Latif University, Khairpur. Did you listen my voice? Hello, I am clearing.
Yes, sir. Yes, you are clear.
Uh, Madam
Yes, sir, you are
Shah,
audible.
in 2006, the institute has six divisions. One is the plant protection division, and date palm production division, tissue culture and biotechnology, and DNA fingerprint. And one of the guy asked him the this is the old institute the people don't know about. You can search about the Shah Abdul University Khairpur website. The hundreds of the papers you can research paper from the website. And here is only the one institute in the university, and the other is the not and the particular on the date palm. And in the Sindhu government has also established the date palm research station at Kodriji, and other in the whole Sindh is not a particular institute of date palm. And if you look into the institute, the Kingfisher University has a date palm research center of excellence, and other the UAE is the Palm Research Institute.
Thank you so much, sir, for answering. Here is someone asking how to eradicate palm bevel effectively. Actually, I think it's
Palm bevel,
always.
madam, this palm bevel is other is a very big issue in the world. And simply, palm bevel, the growers applied the phosphotoxin tablets in the hold to kill the larvae of the red palm bevel. It's a control. It's a management, not a control.
Okay, sir, thank you so much. Here is next question. Which vector cause spread of pathogen in date palm?
Which vector cause spread of pathogen in dead palm? It's a weevil, red palm weevil, enter, and then the second infection of the fungi.
Okay, sir, thank you so much for answering. Guys, I think questions are ended because till now not further question in the chat. If you have any, please write as soon as possible so sir can answer.
Thank you, madam. I enjoy your session.
Thank you so much, sir. It's also a pleasure for us to have
Thank
you
you
with
for
us today.
your presentation. Thank you. Thanks a lot.
Okay, Miss Ikra, are you there?
Yes. Should I conclude the
Uh,
session now?
uh, yes, please conclude the session and give a warm thanks to our speakers.
Okay. Thank you to our audience for the thoughtful questions and also to our speakers for their clear and insightful responses. Once again, we extend our sincere gratitude to Professor Dr. Wilson Thau Yong and Dr. Wazir Ali for sharing their valuable knowledge and expertise with us today. This brings us to the end of today's session. We truly appreciate everyone for joining us and being part of this important discussion. Your attendance will be marked as you have written your names in the live chat. Thank you, and we hope to see you again at our future Vita Nova events. Assalamu alaikum and take care.
Thank you, madam.
Thank you so much, sir.
Thank you, madam. Thanks a lot.
Allah Hafiz.
Yeah, thank you, everybody.
Thank you, Dr. Wilson, too. Okay, Mansoor, are you there? Then please end the session.